So, so good afternoon, everyone, and thank you all for being here. Firstly, I would just like to thank SMMR for giving me the opportunity to present my research today. Now, aquaculture is one of my biggest passions, and with the growing demand for seafood, the industry needs new, innovative technology to to steer its growth in an environmentally and economically sustainable direction. Aquaculture farms face many threats, and one major contributor to fish mortality and economic loss is harmful algal blooms. So, this brings me to my research question. Fluorescence-based methods in the monitoring of phytoplankton communities can aid in the management of harmful algal blooms in aquaculture. So what are harmful algal blooms? Well, harmful algal blooms are the proliferation of microscopic algae or cyanobacteria concentration, which can negatively impact human health, marine life, and coastal ecosystem. It is impossible to foresee these events due to the lack of a well-established and efficient system for the monitoring of poisonous and blooming species in our water bodies worldwide. For instance, in the Gulf of Mexico, annual blooms of the dinoflagellate Carina brevis, a red tide alga, have caused massive disruptions to the fisheries, tourism, the aquaculture sector, and the wider environment through the production of potent neurotoxins. Some major characteristics of algal bloom are they have extremely high algal cell density, which can be greater than 20,000 cells per milliliter. These blooms can occur in fresh water as well as the marine environment and can be dominated by a single or few algal species. Now, these blooms tend to accumulate on the surface, giving the water a green, brown, or red appearance. Algal blooms have developed into a global concern over the past few years. Today, the global impact has affected aquaculture fish and shellfish industries, leading to a yearly economic loss of approximately 8 billion US dollars. The impact of these blooms is not often quantified, except in cases where they have resulted in massive mortalities of cultured animals and significant economic losses. Now, the world map shows the location of bloom events that have affected aquaculture facilities within the last decade. So, for example, if we zoom into the United Kingdom, we can see the impact of these harmful algal blooms on UK finfish and shellfish industry. As seen, majority of the impact were located along the western coast of Scotland, followed by a few bloom events affecting the south coast of England. Currently, there are 8 main genera of algae that are responsible for shellfish toxicity, which are present in the UK waters, with the most common being Alexandrium species, Dinophysis species, and Pseudonychia species. Basically, with the increase in the frequency and intensity of harmful algal blooms, it is safe to say it is a threat to food security. Today, almost 50% of the world's seafood consumption is produced by aquaculture. The growing demand for seafood and the constraints the industry faces based on harmful algal blooms can lead to a drop in the production of an important protein source. Harmful algal blooms are a serious concern, but one of the main questions is, how can these blooms affect culture animals? Well, there are several mechanisms by which harmful algal blooms can affect culture animals and impact on the aquaculture and fisheries industry. Firstly, by the rapid proliferation of non-toxic producing algae that results in the discoloration of water and produces excess foam can lead to mortality by reducing the dissolved oxygen in the water column. Secondly, the microalgae can mechanically damage the gills and disrupt the respiratory function in fish, leading to necrosis of the epithelium and hyperplasia, resulting in mortality from blood hypoxia. For instance, one study reported the impact of microalgae cells on the gill epitheliums of sardine caused by the clogging of the secondary gill lamella, resulting in mortality due to suffocation. Also, various toxins produced from certain algal species can alter the osmoregulatory capacity in fishes. For example, one study by Aguilera et al. In 2016 demonstrated the increase of blood variables of sodium, potassium, and chloride ions in salmon smolt when exposed to various concentrations of Alexandrium cotinella biotoxin. Also, the toxin produced from certain algal species are taken up in the food chain and stored in the digestive glands of bivalves. Now, this can be transferred to other organs by the bloodstream and may increase the toxicity of the shellfish. This can lead to neurological and gastrointestinal disorders in humans, resulting in a significant public health crisis if consumed. And finally, certain toxins produced from cyanobacteria blooms can affect the liver in fish, triggering inflammation, tissue necrosis, and oxidative stress, resulting in mortality. Now, these are some of mechanisms by which blooms affect culture organisms. There is a distinction between blooms causing fish kill and those leading to shellfish toxicity, both of which have a unique ecological consequences. Now we'll look at some of the most iconic bloom events and their impact on the aquaculture industry. Firstly, one of the biggest challenges to the salmon industry in Chile is harmful algal blooms. In 2016, a dictyophyte bloom was responsible for the largest farm fish mortality recorded, with losses amounting up to $800 million in the Chilean salmon industry. Now, this bloom killed a record amount of approximately 40,000 tons of salmon, equivalent to 39 million fish in only 2 weeks. Examination showed that the gills were the most affected organs with significant tissue damage and circulatory disorder. It also noted that during the bloom event, the dissolved oxygen was maintained above 4.5 milligram per liters. However, the fish were noticed to be piping. Therefore, even though oxygen was readily available in the water, due to the damaged gills, they were unable to uptake the necessary oxygen, which led to suffocation. This highlights the devastating impact of algal blooms on the salmon population, as even with sufficient dissolved oxygen level, the fish were unable to survive due to their damaged gills. The rapid and widespread nature of the bloom resulted in a significant economic losses for the Chilean salmon industry. Secondly, fish farmers along the coast of northern Norway experienced sudden mortality of Kate salmon from mid-May to mid-June in 2019. These mortalities were caused due to a heptophyte bloom, and again, fish death was relatively sudden, with gill damage most frequently observed. Upon further analysis showed that there were little or no changes seen in the pathology of the internal organs. This bloom caused a massive mortality of farm salmon, resulting in a loss of approximately 13,000 tons of salmon, equivalent to 7.5 million fish. This loss represented about 2% of Norway's annual farm salmon production and 6.5% of the annual production in Nordlands and Troms. 14 companies were affected, with losses ranging from 5% to 95% of their total potential harvest, and the economic consequence were estimated to be approximately $280 million, significantly impacting the local economy and Norwegian fish farming industry. The salmon mortality typically occurred when the bloom cell density exceeded 2 to 3 million cells per liter. And basically, farm salmon in pens are at much higher risk than wild population, as they can't escape to deeper waters below the bloom layer. To this date, this event was recorded as one of the largest fish kills caused by harmful algal blooms in Norway. So now, if we look at the shellfish industry, harmful algal bloom events have occurred repeatedly in St. Austell Bay and Lyme Bay shellfish sites in the UK since they opened in 2010 and 2015 respectively. For example, throughout the summer of 2018, a phytoplankton community was observed at 2 rope-grown mussel farms along St. Austell Bay and Lyme Bay. This bloom was dominated by 2 species of dinoflagellates, including Dinophysis species and Carenna makimoti. The Carenna makimoti bloom occurred over 2 weeks in August and comprised up to 88% of the standing phytoplankton biomass in St. Austell Bay, whereas the Dinophysis species also bloomed from May to September constituting up to 28% of the phytoplankton biomass. Now, these dinoflagellate species were responsible for the economic loss of 1 million Great British pounds, and upon analysis, the data showed that the accumulation of toxin exceeded regulatory limits, resulting in an 18-week harvest ban. Similarly, in 2017, a large dinoflagellate bloom in Walkers Bay, South Africa, impacted 3 land-based abalone farms, resulting in the death of several million animals. Now, this bloom was dominated by 2 dinoflagellate species, and toxin concentration were found to be highest in the gills, which showed severe and generalized disruption of the gill epithelium, characterized by degeneration and necrosis. And finally, harmful algal blooms of toxin-producing microalgae are a recurrent issue that affects the Chilean bivalve industry. For instance, in northern Chile, the industry has largely focused on scallop production, and in 2016, a dinoflagellate bloom was responsible for a massive mortality of culture scallops, mainly sea scallops with approximately 12 millimeter in shell height. Now, mortality was a result of oxygen depletion caused by the rapid growth and decay of the dinoflagellates, leading to a decrease in the dissolved oxygen level in the water column. Similarly, in this area as well, there is a high frequency and recurrence of diatom blooms caused by the genus Pseudonychia, with at least 2 events documented per year. Now, these blooms have caused multiple toxic events due to the presence of domoic acid, a toxin responsible for anemic shellfish poisoning. Or compared to the southern Chile area, including Patagonia, where harmful algal blooms are caused mainly by Alexandrium cottonella, the dinoflagellate associated with paralytic shellfish poisoning outbreaks. These outbreaks have been documented since 1995 and have been a recurring problem. However, anemic shellfish poisoning events are also a recurring problem in the inner sea of the Los Lagos region, which is responsible for 95% of the national production of the Chilean blue mussel. These blooms have led to harvest bans due to toxin concentration exceeding regulatory limits, and this highlights the delicate balance of the ecosystem and the impact of algal blooms on the Chilean bivalve industry. Basically, every coastal country in the world is potentially affected by harmful algal blooms. There are various programs adopted by different agencies to aid in monitoring and managing of these blooms to mitigate their effects. For example, the Intergovernmental Oceanographic Commission Harmful Algal Information System consists of access to information on current use of taxonomic names and of harmful algae and location of their bloom events. These agencies use satellite images and sample collection to monitor bloom species and toxin concentration, providing a database for bloom events. However, due to the complex nature of algal blooms and their unpredictable behavior, new technologies are needed to understand and manage harmful algal blooms in the marine environment. So, based on these findings, one way forward can be through fluorescence analysis. Fluorescence is a technique commonly used in water science because it offers fast and sensitive optical detection for the monitoring of various compounds in real time, providing a non-invasive approach. Spectroscopic techniques are used as an alternative for algal analysis through fluorescence monitoring by the excitation of fluorescent pigments in phytoplankton. These atoms and molecules absorb light at a specific wavelength and emit light at a longer wavelength. These fluorophores generally contain pi conjugated systems that allow for the absorption of ultraviolet or visible radiation. And in this process, the electron that obtain energy transition from a single ground state to a singlet excited state, as described in the Jablonski diagram to the right. And fluorescent emission occurs when electron returns to the electronic ground state. Now, fluorescence spectroscopy is a powerful tool that can be used to discriminate between major phytoplankton groups. Compared to traditional methods used in harmful algal blooms monitoring, which requires sample collection, expensive equipment, and technically trained staff, these may include morphological-based analysis or molecular techniques. Microalgae contain a diverse variety of pigments such as chlorophyll, carotenoids, and phycobiloproteins, whose unique composition provides a mechanism for monitoring. These pigment-based approaches have proven to be a promising tool for in situ exploration of bloom composition. And with the advancement in fluorescence detection technology, researchers have turned to classifying algae based on the fluorescent properties of these photosynthetic pigments. Therefore, fluorescence can be a viable tool for algal analysis through specimen concentration and classification prediction. Now, the excitation emission matrices obtained from fluorescence spectra can provide a comprehensive picture of the fluorescent properties of various samples and obtain abundant spectral fingerprint information. These fluorescent pigments have specific excitation and emission wavelengths, and the emission excitation spectra for a given fluorophore are mirror images of each other. Usually, the emission spectra happens at a longer wavelength than the excitation spectrum, and these type of spectrums are used to observe changes in samples where the intensity and peak wavelength of a spectrum may change based on factors like temperature, pH, or even concentration. For instance, tryptophan is excited at 280 nanometers and emits at 300 to 400 nanometers, as compared to chlorophyll A, which is excited at 400 to 460 nanometers and emits between 650 to 730 nanometers. Fluorescence analysis has been applied to various applications in monitoring blooms, such as for bloom concentration, in which chlorophyll concentration is a representative of the state of the water quality and has generally been used as a key indicator for measuring algal blooms. Spectrophotometry is a classical method used for determining the quantity of chlorophyll in surface water. Now, the ability for chlorophyll to fluorescence is the basis for all commercial fluorometers and the development of in situ probes used for monitoring microalgae from its chlorophyll A pigment. Compared to algae classification, where several studies have demonstrated that the classification of phytoplankton can be achieved using 3D fluorescence spectroscopy under variable circumstances. For instance, one study by Zhang et al. Classified 11 different microalgae using their 3D excitation emission matrix in combination with PCA, to analyze these algae. The spectral differences observed between the species with distinguishing peaks at different excitation frequencies and intensities were the basis on the algae classification. Similarly, Zhao et al. Analyzed 12 microalgae species from 5 classes and compared the results with the emission spectra of chlorophyll A at 685 nanometers. The analysis showed a high level of accuracy for algae classification. Now, these studies demonstrated the notion of using fluorescence analysis as a tool for the classification of algae at the division level in both simulated and in situ environments. This technology has benefited in the monitoring, but is insufficient for long-term monitoring system that requires species identification and concentration estimation. Although phytoplankton can be separated into different classifications using these tools, future research should focus on developing methods to effectively differentiate microalgae species to improve monitoring accuracy for the aquaculture sector. Moreover, for species identification, the rapid and accurate identification of the causative species, particular toxic species, is of greater importance for better management and controlling harmful algal blooms. For instance, it was demonstrated by Yoshida et al. The application of monitoring chlorophyll fluorescence at the wavelength of 670 and 690 nanometers can aid in identifying specific algal species. The authors evaluated the spectral shift of chlorophyll between the wavelengths and developed an identification sensor. While they successfully used this method to differentiate between two specific algal species based on their fluorescence, it could not identify all the species in the study. Furthermore, Shan et al. Also used fluorescence spectroscopy in combination with discrete wavelength transformation analysis to identify specific peak characteristics for 8 microalgae species. All 8 species could be differentiated with discriminative probabilities greater than 80%. The pure sample identification accuracy was over 95%, whereas dominating algal species in mixed samples had an accuracy rate of 77.4%. Now, these studies have illustrated that the spectral information obtained through fluorescence analysis could provide detailed features of various pigments that can be utilized for harmful algal blooms species identification. However, the spectral analysis of certain fluorophores in microalgae are limited due to the spectral similarities between species, which hinders the accuracy of identification. Hence, the fluorescent properties of protein-like peaks can offer an alternative to monitoring blooming species. Now, these organic substances are produced from microalgae in varied forms and quantities. Algal organic matter is comprised of a heterogeneous mixture of dissolved organic molecules, such as biopolymers, proteins, polysaccharides, lipids, and nucleic acid. Furthermore, during bloom events, there is a simultaneous increase of dissolved organic carbon and cell concentration associated with the release of algal organic matter, which can aid in algal monitoring. Spectroscopic techniques like fluorescence spectroscopy is the second most abundant technique used for the analysis of organic matter when compared to dissolved organic nitrogen analysis, hydrophobicity analysis, and high-performance size exclusion chromatography. Several research has showed that protein-like peaks are produced during the growth phases of algal bloom. Now, these research articles provide an important insight into the aromatic algal organic matter compounds by utilizing fluorescence spectroscopy. The authors highlighted that fluorescence spectral analysis of algal organic matter during blooms contain protein-like and humic-like substances such as tyrosine and tryptophan. In the marine environment, algal organic matter accumulation varies based on the particular blooming microalgae species. Moreover, the identification and classification accuracy are hindered when there are multiple dominant microalgae species present. Therefore, the fluorescence information obtained from only a few sources is inadequate for accurate identification. Thus, the several protein-like peaks produced during the growth phases of blooms can be employed for further monitoring of bloom species. By using the fluorescence signatures of algal organic matter, it can be possible to potentially discriminate across the complex pigments of various microalgae species. Now, in establishing an efficient management tool for limiting damages caused by the occurrence of harmful algal blooms, the creation of an early warning system should be the first step. Close monitoring and early warning research are essential to measure and mitigate the impact of harmful algal blooms on the aquaculture sector, providing a timely response to counter emerging threats. Obtaining early warning of harmful algal blooms is highly valuable to the aquaculture industry. However, it is a complex process due to the fact that different algal species have varying life cycles and toxicity levels. Furthermore, the location and timing of harmful algal blooms are influenced by local oceanography. The application of fluorescence detection methods can provide a dynamic approach to the development of early warning systems in conjunction with species identification and environmental conditions. Therefore, it is imperative that instead of taking a reactive approach after the damage has been done, being able to predict a bloom species can aid in early intervention strategies, which can improve the economic turnover in the aquaculture sector. Recently, increasing attention has been geared towards the development of prediction models, which can aid in monitoring microalgae blooms and improve the effectiveness of management programs. Machine learning models are utilized due to their ability to handle complex, often non-linear and noisy datasets, which can produce highly accurate predictive models. For example, Lee et al. Developed a model utilizing a multi-label convolutional neural network in combination with fluorescence spectroscopy to identify marine microalgae. The authors highlighted the potential of CNN as a promising tool for the precise identification of marine algae. Similarly, Shin et al. Demonstrated that recurrent neural network was the most effective algorithm in the prediction of chlorophyll A when compared to linear regression models. Overall, neural network models can effectively extract spectral features for fast and accurate identification of fluorescent substances. Also, aquaculture fish and shellfish are significantly affected by toxic bioactive molecules produced by marine microalgae. These toxin compounds may increase during blooms and accumulate in shellfish or higher trophic species, leading to a public health hazard when consumed. These toxins can be unique to a species or multiple species of the same genus, and they can exhibit varying toxicity levels due to geographic location, seasonal environmental factors, and even daily weather conditions. Of the various species of hazardous marine microalgae worldwide, dinoflagellates make up 75%. Many species of dinoflagellates can generate a variety of toxins, and marine toxin-producing species are remarkably diverse and are highly dangerous at exceptionally low concentration. Therefore, toxin monitoring programs are essential in areas where harmful algal bloom events have become prominent. Hence, it is crucial to understand how environmental factors affect the development of toxin bloom and the production of toxin in the marine environment. So based on these findings, the proposed methodology of my research will be to identify potential algal species that will be purchased and cultured under biosecure protocols for analysis. These cultures will be analyzed using fluorescence spectroscopy to identify various fluorophores, which can be used as a fingerprint for the species. Also, PowerFact will be used because it is a well-established method for the analysis of excitation emission matrix spectra. It allows for the decomposition of the multi-way data, focuses on the features of interest, and produces a distinctive illustration of the results. It also offers a more in-depth exploration of the extraction of the spectral data information. Also, the microalgae toxin will also be monitored throughout the growth cycle to evaluate the stage of growth and what conditions trigger toxin release. And finally, a predictive model will be developed using the excitation emission matrix to identify the microalgae species. This model will be tested using mixed algal samples and water samples collected from aquaculture facilities. So overall, the main steps of this research will include data collection, which involves culturing microalgae species and obtaining spectral data using fluorescence spectroscopy. This is then followed by data analysis, pre-processing, and PowerFact decomposition, which will be used to remove outliers from the spectral data and produce different spectral components. This data will be used in combination with machine learning algorithms to develop a predictive model. This model will be trained, tested, and validated before being deployed. And the output of the predictive model will be able to identify microalgae species and bloom concentration, therefore providing sufficient information to aquaculture farms, allowing them time to implement mitigation strategies to reduce the impact of algal blooms on their facility. So in conclusion, harmful algal blooms pose significant challenges to aquaculture and fisheries resources, causing substantial economic losses and potential food shortages. Identifying species in seawater is complicated due to the complex pigment distribution, but fluorescence information from various detection channels can provide accurate results. Early warning of algal blooms is crucial to farm operators as it allows them to employ management practices to minimize the reduction of mortality, hence improving the economic turnover. These models should offer precise and quick responses appropriate to the affected area. Furthermore, it should be based on a comprehensive understanding of the biology of the causative microalgae species and how it affects cultured organisms. Therefore, it is crucial for the management of harmful algal blooms that research is continued in the development geared towards enhancing models that can forecast and predict species responsible for harmful. This technology can proactively provide steps towards managing this threat, leading to a better protection of aquaculture resources and ensuring its sustainable future. Well, thank you everyone for listening.